It's indeed a pleasure now for me to introduce Peter Marks, who is our keynote lecturer. Peter received his PhD in cell and molecular biology, as well as his MD from NYU. He did his internal medicine residency and Hem-Onc clinical training, fellowship training at the Brigham and Women's Hospital, where he joined the faculty and served as clinical director of hematology before taking some time moving over to industry for drug development, and then coming back to assume several leadership positions at Yale before being recruited to CBER in 2012 as deputy director, and he became center director in 2016. Those of you who work in industry or even just watch the news know what is being put on CBER's plate right now. And I can think of no person with a better skill set to handle the opportunities and challenges moving forward than Peter Marks. And we're very fortunate to have him here to speak on Bespoke Therapeutics. Peter, turn it over to you.
So what we're going to do, what I'm going to try to do for the next 14, 15 minutes is take a vacation from COVID-19 and to talk about something that's very near and dear to my heart, which are bespoke therapeutics. For those who are new to this concept, these are really individualized therapeutics meant to treat handfuls of patients rather than large numbers of patients. So I want to talk a little bit about the need for these products, talk about the challenges that can occur in product development, and talk about some of our efforts to facilitate this. I apologize to anyone who's heard me talk about this before. Hopefully you'll see a little bit how my thinking has evolved a little bit. So for those just to understand the paradigm of one of these therapies that's actually, this is one that's used not in just handfuls, but it's a good model of what can happen if you have a very effective gene therapy. Onasemnogene, or Zolgensma, is a therapy for spinal muscular atrophy. It was developed for spinal muscular atrophy and approved for essentially type 1, which is a very severe disorder, basically takes children's lives by about 3 years of age. The development of this therapy was a very nice advance for systemic gene therapy. It's the first and right now the only systemic gene therapy that's directly administered intravenously that's approved. The data on the left side of this slide shows that really the great majority of children treated early with this therapy end up being normal over the course of several years. And the child on the right was a girl that was brought down to Capitol Hill and had senators crying because of the success story of taking a child that would have been either on a ventilator or dead at this point and having them, for all appearance, to be normal. Now, there are some uncertainties here. How long will this last? How far can this paradigm be expanded? But at least for now, this is very exciting. And obviously, AV, adeno-associated viral gene therapies, have their challenges, but the gene therapy paradigm is here to stay. So the importance of these therapies for disorders that are very rare, perhaps even rarer, spinal muscular atrophy, is that in aggregate, there are thousands of them. And right now, there are probably hundreds that could be addressed by gene therapy. It's just that they're for such small populations that we don't have a way of going through the development in a facile way. Getting to paradigms that'll let us do this is probably pretty important because a couple things. One, if you think about taking even somewhat more common diseases, when you start to address problems with gene therapy, particularly if you're going to use genome editing with CRISPR-Cas9 and you're going to address molecular defects, more common diseases become actually a host of rare diseases when you look at the molecular genetic level. And the other thing is that if we finally start to have high throughput of developing gene therapies, we will probably start to develop the knowledge of efficiencies that will allow us to have the insight we need to make gene therapies for larger populations. And just to take a digression here into what we're really talking about. So personalized medicine was all the rage at the turn of the millennium, which seems a long time ago now. And it's moved now. I think we're thinking here, aside from, we don't get me wrong, we still want to find the right drug off the shelf to treat patients when we can. But increasingly, we're talking about creating the right drug to treat the patient, or individualized medicine. And individualized medicine really breaks down into two paths, one of which we're more familiar with at FDA right now, which are customized products, which are products that have the same indication, the same mode of action, but might have some distinct property that makes each individual product slightly different from another product. An example were personalized cancer vaccines. And there are things you can imagine that if you had pulsed dendritic cells, those would be pulsed with an individualized peptide mixture from somebody's tumor. That would be a customized product. But then there are also created products where there'll be a different indication, different mode of action, but they may have kind of a device-like feel to them, where you're using the same part of the same thing over and over, but essentially just iterating the same genetic backbone, for instance, for a gene therapy vector that you're putting in different inserts into. And there, an example might be, gene therapies for two different hemoglobin mutations using the same vector backbone. And that could, those that you can imagine could just be very slightly different to deal with different mutations. And this kind of boils down to, personalized medicine was ready to wear, customized therapies were kind of made to measure, where the suit's cut, but the seams are left open. And really, the bespoke therapies, or these created products, are what we're talking about. And the challenges here are ones of manufacturing, non-clinical development, clinical development, and also product access. So for manufacturing, this is a slide that I've shown before. We really have this issue, and it's funny because it kind of goes, we were talking about real world evidence not being at the extremes and not in the center here. The issue is that for larger products, we have manufacturing in place, not giant populations, but for modest-sized populations, we're pretty reasonable for manufacturing technologies for gene therapies right at the moment. Clearly, we need to do better, but at least we can do this at a cost, at a way that they're being made. For small runs of products, runs where you're talking about treating a few dozen people, the cost right now has become such, and the availability of capacity is such, that it's really limiting development. And it may be that, without going into, this could be a talk in and of itself about manufacturing. It may be that the way we're going to get there ultimately is by getting to the point of having really devices that can actually do the manufacturing here. And although that sounds, somewhat futuristic, I don't think it's as futuristic as it might seem. And it's probably somewhere maybe a decade off. But we probably need to be thinking in this direction. Sorry, this is one of my son's favorite machines when he was little, in various places. The infinite way of mixing and matching here. But this concept though of a device is something where you wouldn't have to have the incredible setup costs. You could imagine that you might be able to have a set of backbones that you then put different inserts into, where we could be expediting that process. The other issue, non-clinical development for products. So we know already that animal models may be less than ideal for certain things, particularly as we're starting to think of genome editing, because no matter how much you might like to think, no matter monkeys and humans don't share the exact same genetic sequence, and certainly mice and humans, although they may be 99+% identical, are not identical enough in their in the places that you needed them to be able to use them as perfect models for genome editing. So we need to think about other solutions, whether they be human organoids or humanized mice. Those are things that might help with safety testing. For clinical development, we sure need help here. How do we appropriately document natural history disease or collect baseline data? How do we deal with efficacy in very small populations? And that may be by starting to have templates for collecting baseline data. It may mean that we need to make more readily accessible to rare disease communities, as they already are. They already are partially accessible, but not perhaps accessible enough. These abilities for patient communities to collect data in a way that will actually be meaningfully useful for subsequently creating drugs. And also by using Bayesian clinical trial designs, this concept of maybe each patient helps you gain more information, and you essentially iterate on the model so that at a certain point, after you've treated a certain number of patients, you decide that the likelihood that the next patient is going to respond is sufficiently high that you can consider the drug approvable and or reimbursable. And then obviously there's product access, which is whether it's under investigation new drug application or a biologics license application. The issue of having continued access for patients to these products is potentially prohibitive. And this has become, I get probably one email a week to every other week of a patient group that's looking to have a gene therapy applied to their rare genetic disease. But when they go to a contract manufacturer with their academic partner, the cost is just so large that even their GoFundMes can't get there. And so how could we get there? Well, one of the things we've been thinking about is whether public-private partnerships could at least start to put together the playbook that's needed here. And could you potentially put together a playbook, something like the cookbook or Maniatis of gene therapy, that lets people understand what you need to do to get there to reduce the cost and to increase the efficiency. And with the Foundation for National Institutes of Health and the National Center for Advancing Translational Sciences, we're working together to try to put together a few test cases where we may use the same vector with a commercial manufacturer, perhaps, who's used that vector before, use that same manufacturing process to get through a couple of examples, allow those to get into clinical trials, and try to get the data, try to then put in the regulatory framework here that allows one to leverage applications so that, again, reducing the regulatory burden, and by trying to essentially optimize each of the pieces of the puzzle that go together to get something from a concept over to a therapy that helps a patient, try to enable this to happen in a way that allows these to become accessible for handfuls of patients. This kind of cookbook for the development and manufacturing of bespoke therapeutics, you might say, well, why do you really need this? People know what to do. But when you actually look from the FDA side, the number of times the wheel has been reinvented is amazing. Now, it turns out you can actually have square wheels that give you a reasonable ride. That is, if you have the correct surface that you're putting them on. But probably it would be easier if we all agreed on what the surface looks like and what the wheel should look like, and then people just keep using the same one. That's pretty much why our axles are still the same length as Roman chariots. So we need to try to help there. And so that's one area. And then the other issue is whether we can help with this regulatory aspect by allowing people to leverage non-clinical and manufacturing data from one application to another. Do we really need to reinvent all the information for a vector backbone over and over again if we can figure out some parameters like what size inserts can it reasonably accept? So really what we'd like to do is to try to help advance these bespoke therapeutics, ultimately knowing that if we can help with these small size products, it's going to, you can you can see maybe I'll be wrong, but I find it hard to believe that if we can do it frequently on a small scale, that we can't develop insights that will really help us for larger populations in a very large way. We'd like to do this we'll help individualize product development. We'll continue to provide input and collaboration on novel endpoints and encourage innovative clinical trial designs, all in this hope of seeing gene therapy become a reality. And I'll stop there.
Thank you very much, Peter. I just want to congratulate you on a terrific lecture. I'll start. There haven't been any questions. Please send questions. We've got about 3 or 4 minutes for questions. I'll start off by just saying that, Peter, with the enormous growth in what CBER is charged with, cell and gene therapies are exploding, not to mention vaccines and everything else you're doing. How do you, as a relatively smaller center, garner the clinical expertise needed to make decisions? I'm sure you have CDRH and CDER people at your disposal, but could you just walk us through some of the challenges with the incredible growth at CBER with regard to just having the clinical expertise in-house to answer important questions in the review process?
Yeah, no, thanks for that question. We have a pretty diverse group in our Office of Tissues and Advanced Therapy. So we have a fairly diverse number of medical officers, some, obviously, a fair number of hematologist oncologists, surgical oncologists, neurologists, and we will have to grow that diversity significantly over the next few years as we ramp up. But we also obviously do pull on the other centers, and obviously we collaborate with the Oncology Center of Excellence very extensively when it relates to oncology products. And we have to have a very vigorous relationship with CDRH, because a lot of these gene therapies now are being delivered with very novel devices. One of the characteristics of our center in general has been we're a nexus for a lot of collaboration within the FDA, because we have touch points with CDRH, with CDER, Center for Veterinary Medicine, because a number of our cell therapies or even some of our products come from genetically altered animals. So we have that. And it turns out we regulate a number of products that also can sometimes be considered foods. So we actually interact with CIFSAM as well. So that's one of the fun things about being at the Center for Biologics.
And we have time for, I think we have actually just a comment on the chat, but no questions. So I'll ask one more. We have about a minute. What keeps you up at night? You've been in the news every day almost, and so much going on right now. When you go home and try to unwind, what do you worry about with regard to the center and with regard to the health of our public in general?
So now I'm going to have to come back to COVID-19. I think I worry about two things with COVID-19. One is that I think although this virus doesn't seem to be mutating quite like influenza virus does, I am worried that given the number of hosts that it's in and given where it's headed, that before we actually get good control, we could actually see it mutate out into something that could escape some of our vaccines. So that gives me that keeps me up at night. I'm sure as heck, I'm not trying to scare anyone, but I have to be in the back of my mind that we have to be thinking about that sufficiently such that we are already starting to think about what we'll do if that happens. And the other piece that keeps me up at night, and maybe it's just me, is that given that we have these incredibly effective vaccines, that we've really been lucky. I think when I saw the percent effectiveness of the mRNA vaccines in older people, 75-year-olds, at the rate they are for COVID-19, that's tremendously exciting. Given that we have these, how are we going to make sure that we ramp up? To me, it's almost inexcusable that we don't have incredible capacity at this point to pump those vaccines out. And it keeps me up at night thinking that we may end up having to deploy inferior strategies it keeps me up at night thinking we might give people one vaccine and give them 50% or less effectiveness when we have at our disposal potentially a vaccine that is super super in terms of what you might see in an older population. I'm not trying to oversell that, but a vaccine that has 95% effectiveness in 70-year-olds, that's pretty amazing, because most vaccines, if you look at flu vaccine as you get into that age range, it's nowhere near that. So those are things that keep me up at night, that where this pandemic might go, and our limitations perhaps, which I don't know whether they're partially artificial and partially real, about being able to ramp up to address them. I mean, we do in the United States have the capacity to ramp up production. We know we did it in the Second World War, from going to making nearly no airplanes to making close to 20,000 airplanes in a year. We may need to do something similar here for our manufacturing capacity.
Peter, thank you for your wonderful comments and for joining us today, and for all the work you do on behalf of our public.